Okay, so welcome everyone to another Mercodia webinar. And this time we are very glad to have with us Professor Fredrik Bäckhed. His research combines clinical-oriented research with non-probiotic mouse models to address the role of the normal gut microbiota in cardiometabolic diseases. And maybe many of you don't know, but Professor Bäckhed, he holds a PhD from Karolinska Institute in Stockholm, Sweden, and performed his postdoctoral training at Washington University in St. Louis, where he identified the gut microbiota as an environmental factor that regulates adiposity and obesity. Nowadays, Professor Fredrik is a professor at the University of Gothenburg, and also he's appointed professor at the University of Copenhagen. He has been guest professor at the University of Oslo and recently did a sabbatical at University of Hawaii. So this is just a brief introduction. I would like to mention to all the attendees that if you have questions, please feel free to write your questions in the space that is shown, and they will be read at the end of the presentation. So Professor Bäckhed could reply and give a good answer to them. And at the same time, again, if you have any further questions, don't hesitate in contacting us. So after this brief introduction, Professor Bäckhed, it's all yours.
Thank you so much, Miguel. It's really a pleasure and privilege to be here and present some of our data here today, and also give you an introduction to the gut microbiota, this beautiful world of microorganisms that lives in our bodies and on our bodies, and how they can affect us in health and disease, and where we are right now, at least in our view in the field. So I would mainly speak about the gut microbiota and diabetes and other cardiometabolic diseases. But of course, it's a really big buzz today to talk about the microbiome and what it may do and it may not do. And just because you have a very hot field today, it doesn't mean it's new. So this is Antonio van Leeuwenhoek, who invented the modern microscope. And he used that microscope to look at what he had in his oral cavity and in his feces. And he could then describe that there were small molecules, but they seemed to be alive. And they were those that were very, very tiny, those that are larger, spirochetes, those that are motile, and some coliforms. And since he didn't know what they were, and they were small like molecules, but seemed to be alive, he called them animalcules. And then we have sort of improved our way of imaging these. And today we can use scanning electron micrographs to really depict the microbes here. Here you have a pseudocolored microbe sitting on the epithelium of a mouse colon, embedded in the mucus here in green, and that is exposed also to some food particles. So this is really the trifecta we want to explore in our group, to understand how the microbes interact with us as human beings or the host, as well as with what we eat and provide us and the microbes with. As a brief introduction, and many of you probably know this, is that we have actually more bacterial cells than human cells in our body. And you will hear estimates that we may be 10 times as many bacterial cells as in human cells in our bodies, or at least maybe 1 or 2-fold more. So this means that we are actually more bacteria than human beings on a cellular basis. But since the bacterial cells are very small compared to human cells, we still are, of course, most of our cells we see are human cells. And since each of the microbe has its own genome, we have many more microbial genes than we have human genes in our holobiont or meta-organisms. So for each human gene we have 1,000 bacterial genes, which means that we are on a genetic basis 0.1% human and 99.9% bacterial. And we think we should really view the bacteria in our gut as an organ on its own right that consists of 500 to 1,000 different cell types. They have a biomass of about 1 to 1.5 kilograms. So it's almost the size of the human liver. And of course, these bacteria can change during our lifespan, and they will can affect our physiology. But when we talk about the gut microbiota, we seem to often including us, forget about that we have actually different kind of habitats. Just like on Earth, we have different habitats. In the gut, we do have a ranging environment from the stomach, which I could view as a desert, where we have very few living organisms. Those that are living there may be Helicobacter pylori, that is here like the palm tree. Then when we go further down in the gut, we come to the duodenum, which is a relatively harsh environment, few microorganisms, a lot of different enzymes, and bile, etc., that will keep down the number of bacteria. When we get down to the ileum, we have maybe 10 to the 8th organisms per gram content. I would view that as a Swedish nice pine forest. So we have some biomass there, but not that much until we reach the colon, where we really have a rainforest. We have a vast diversity of microorganisms, and we view them as a bioreactor that they can really degrade and consume a lot of the nutrients that we eat, that our body cannot themselves digest. So the microbiota has been associated with many diseases, and the key word here is it's been associated. So basically, scientists have been seeing a link between a given disease state and an altered microbiome. And you will see them here. And some are also syndromes like ASD, so autism spectrum disorders. We see links between the microbiome and like asthma, dermatitis, but also, of course, then metabolic diseases such as type 2 diabetes, obesity, NAFLD-NASH. But in most instances, there's not been a causal relationship. Some has been proven to have a causality if we go to model organisms, but we still have to really translate these findings into new therapies that are based on the microbiome. And I hope that is something we may be able to contribute to it in the next couple of years. So one big difference between us, human beings, and the microorganisms that we carry is that our number of cells and are relatively constant throughout life. In contrast, the microorganisms will vary over our lifetime, depending on many different factors, some obvious ones like antibiotics, but also other pharmaceuticals, such as diabetes medications like metformin, but also statins will have a profound effect on the microbial composition. We see that if we expose mice to cold, they will change the microbiome. And of course, micronutrients from the diet will affect the composition of the microbiota. Especially the dietary fiber seems to be very important to maintain a higher diversity of the gut microbes. But especially the macronutrients that we eat are not only shaping the microbiome, they will also act as substrates for microbial enzymes, giving rise to microbially produced molecules that then will be secreted out and affect different organ systems in the host. So we thus want to even view the microbiota as an endocrine organ that can have effects regulating our metabolism and our physiology. And we are starting to scratch the surface of the microbially derived metabolites and what they will do. And we will give one such example that we are very interested in the end of this talk. So how can we study causality when it comes to the microbiome? One way of doing it is to use germ-free mice. So these are mice that never been exposed to microorganisms in their entire life. We maintain them in plastic isolators where they are then exposed to sterilized food, water, and we can then introduce either a given microbe or consortia of microbes or an entire ecosystem from a human or from a mouse or another living organism. And we can then monitor the mice and see how these microbes can affect the physiology. So this is what we did when I started my career in this field. And as Miguel was saying in the introduction, I did my postdoc together with Jeffrey Gordon, now almost 20 years ago. And at that time, we wanted to ask a very simple question. Does the gut microbiota contribute to the amount of body fat in the host? And we did that by examining and compare and contrast germ-free mice to conventional raised counterparts. And we found that the conventional raised mice had significantly more body fat compared to a germ-free mouse. And if we took these germ-free mice and reintroduced the microbiota from the conventional raised mice and then reanalyzed them 2 weeks later, we saw that they had normalized the amount of body fat in the body, clearly demonstrating that the gut microbiota can contribute to the amount of body fat. And we could relate this that the microbes that would then digest and metabolize complex fibers, dietary fibers, in a chow-based diet, which is basically only plant material. So if you don't have a microbiota, the mice have to eat more, but they still gain less fat. So we think this may be actually of relevance to humans, but we at that time were more linking it to obesity. But I think we should have rather focused on that this may be one of the more basic foundations for the symbiosis between us and the microbiota, that the microbes actually helped us to rescue calories from the diet when we were foraging and looking for diet as a hunter-gatherers. And it's been estimates now that the microbiota may contribute to between 10 and 30% of energy in the body through fermentation to short-chain fatty acids. But it also spurred us to investigate whether the microbiota could be a factor that could contribute to obesity. So we then switched the model system and we use what we call a Western style diet, which is basically you take a normal mouse and you switch it to a diet where 40% of the energy comes from saturated fatty acids and 40% from sucrose and 20% from protein. We then monitor the mice body weight for 8 weeks, and we know that the colonized mouse will gain weight. But what about germ-free mice? Will they gain weight or will they remain lean? So we performed an experiment where we took germ-free mice, and we had mice that been germ-free at birth and then been recolonized with a gut microbiota from a normal mouse, and then we exposed them to a Western-style diet for 8 weeks. And what we found by weighing them every week was that the conventionalized mice, those with bacteria, they gained weight and developed obesity, whereas the germ-free mice did not. And gaining 5 grams of weight may not seem much, but if the mice weighs 20 to 25 grams, it's quite significant. So at the time, there was a lot of interest in the gut microbiota and obesity in humans. So several studies came out and said that there is an altered microbiome, but it turned out that the studies were relatively small and it was hard to find a given direction of these. So then Pat Schloss and Mark Zay did a meta-analysis, and what they found is the signal that seems to be consistent over studies is that obese individuals seem to have a less diverse microbiota than lean controls. However, they also pointed out that many of the studies have been underpowered. And I think this is something that is not unique for this field, but also for all the microbiome fields, is that the sequencing has been relatively expensive and computing intensive. So the studies have, in general, been quite small. So what does it mean that an obese individual have a low diversity of their microbiota? It may be as simple as that the diet that an obese individual eats is less complex, less fiber rich, and thus you have a lower diversity. But I would like to highlight this paper from Julia Goodrich when she was working with Ruth Ley, and they had a pretty large study, and they actually identified one bacteria that was associated with a low BMI, and that was a group of bacteria called Cristalliciniaceae. And there's now ongoing studies in humans to see whether a Cristalliciniella could be a probiotic counteracting obesity. So the jury is still out there, but I think this is exciting, and I think it also highlights that we have to go into more proof of concept studies in humans to see whether a bacteria actually contribute to disease. So in our group, we're not that interested in obesity per se, but we're really interested in the metabolic syndrome and the diseases that obesity is associated with. And one of the bottom line hypotheses we had is that, yes, obesity is associated with these diseases, but not all individuals or patients with obesity will go on and develop cardiometabolic diseases. So can the microbiota be a risk factor or one contributing factor for individuals going down this line of this trajectory to type 2 diabetes, but also to cardiovascular disease and maybe even a chronic kidney disease, etc. So we and others, similar to what I presented from obesity, we did cross-sectional studies, taking samples from patients with or without type 2 diabetes, and then profile the microbiome. What we have found in the field of type 2 diabetes is that there seems to be a little bit more of a consensus that patients with type 2 diabetes seems to lose a group of bacteria known as the butyrate-producing bacteria. These are bacteria that can digest and metabolize complex dietary fibers and produce a small short-chain fatty acid based on 4 carbons called butyrate. But it also, there was another paper coming out a few years later that highlighted that one of the caveats by doing cross-sectional studies, taking patients that already developed disease, is that they also will have treatment. So although we controlled for it in this paper here, what Kristoffer Forslund and Ulf Petersen highlighted was that a diabetes medication called metformin has a profound effect on the microbial composition in the human gut. So this has caused some problem for us because people have been saying that the microbiome is not altered in type 2 diabetes. It's you're studying a metformin effect. So we decided that we need to do something about it. And together with Göran Bergström, who's a clinical professor here at the University of Gothenburg in the Wallenberg laboratory, we set up a big study to study how does the microbiome look like in treatment-naive subjects. So this is Göran, and his team has then been sending out more than 26,000 letters to individuals between 50 and 64 years of age in this city, with inclusion criteria that they should not have any known type 2 diabetes, and as such, they should be treatment-naive. More than 5,000 individuals responded to our call, came to the clinic, were subjected to an oral glucose tolerance test. And then we couldn't afford to include all 5,000 individuals. So we set up a subset of these. And we took basically everybody with type 2 diabetes, screen-detected type 2 diabetes, with prediabetes either defined as impaired glucose tolerance or impaired fasting glucose. We have had them to fill out a questionnaire for fine risk score, which is basically identifying individuals with a high risk of developing type 2 diabetes, and also individuals with first-degree relatives with type 2 diabetes. We call them the high risk, but they have a normal glucose tolerance. And then we had individuals with normal glucose tolerance. We have obtained several blood samples for metabolomics and proteomics and other omics that we are processing. We have food frequency questionnaires, and of course we have fecal samples. And as you can imagine, this is it's been a big study taking a lot of time. So we basically decided to make an interim fecal metagenomic study of the first 1,000 subjects. And then we took advantage of a sister cohort that was also recruited here in Gothenburg. So we had another almost 500 individuals to validate our findings. And we performed the sequencing, and we generated a gene catalog with more than 15 million microbial genes. And we then started to analyze the samples. And this has been a really tour de force led by 2 very talented researchers in the group, Tony Wu, who's now an assistant professor at Fudan University, and Valentina Tremaroli, who's our team leader in microbiome research in the group here. So how does this study looks like? So this is basically each individual here is one individual, and they are plotted on the fasting blood glucose versus the 2-hour oral glucose tolerance. So you have those with a normal glucose tolerance, you have those with an impaired fasting and glucose, impaired glucose tolerance, those that have been having detected as screen detected type 2 diabetes based on the fasting glucose. Those have been detected as type 2 diabetes based on the glucose tolerance. And then we have a group here that we think are very interesting, that is those that have both the impaired fasting glucose and impaired glucose tolerance, and we call them then the combined glucose intolerance. In the validation study, we had to skip those with high risk normal glucose tolerance and also the impaired fasting glucose due to just it got too expensive, and we wanted to validate some of the other findings. So to make a long story short, when we looked at the microbiome, and this is a plot that you may or may not be used to see, it's called a principal component analysis. And it basically describes how much of the variation in the microbiome can we explain. On the x-axis is 8%, and on y-axis it's about 4%. The difference along the x-axis is more important than the y-axis. And what we see is that the type 2 diabetes and those with combined glucose intolerance, and mind you, these are untreated individuals, are different from those that had normal glucose tolerance, and that those with impaired glucose tolerance seems to be started to move in this direction. The impaired fasting glucose do not seem to do that much. And when we start to look into what are the bacteria changing, and that's in this heat map here, where we have different bacteria in each line, and the rows here we start with those with a low risk normal glucose tolerance, high risk normal glucose tolerance, impaired fasting glucose, impaired glucose tolerance, combined glucose intolerance, and type 2 diabetes. And the pattern we see is it seems to be a gradual reduction of bacteria with a big change happening here in combined glucose intolerance. So these are not necessarily clinically having type 2 diabetes, but we yet see an altered microbiome. We could then also validate many of these changes in the validation cohort. Fewer bacteria are increased in type 2 diabetes. And I will come back to that. But let's stop for a minute here and look at those that were reduced in type 2 diabetes. First of all, we find that these are the butyrate producers. We see a reduction in butyrate producers also in non-treated human individuals. So I think it's fair to say that the microbiome is altered in type 2 diabetes. It's also altered before disease onset. And we try now to figure out why and how does the microbiome contribute. But if we want to be a little bit more philosophic, one could also view it from the Anna Karenina, that states that all happy families are alike, but each unhappy family is unhappy in its own way. We think that this may explain why we see fewer bacteria being increased in type 2 diabetes. So when we take our microbiome-centric version of this, we would like to state that a healthy microbiome is rich in butyrate producers, whereas a diabetes-associated microbiome can have blooms in different pathogenic organisms or potentially pathogenic organisms. So whereas all of them sort of lose this diversity in the butyrate producers, you may have spurs in different bacteria instead. But since they're different, we may not find statistical significance. That's why it's just few of them that reaches them. And perhaps some of them could then be contributing to disease onset. And we are currently working very hard now to establish this, if the microbiome can be causally linked to type 2 diabetes. So what's the purpose of this work? Well, one of them is, of course, to trying to identify individuals based on the microbiome that may be at risk of developing type 2 diabetes. And could we then targeting the microbiome? And this is, of course, a lot of hand waving. We're not there yet. But could we make personalized diets? Could we supply basically patients or individuals at risk with a probiotic based not something that is available on the shelf today, but from bacteria we isolate that are at a lower abundance or basically gone from those that develop a disease. This is something we're doing right now. We're very excited about it, and we are currently performing one such intervention study with a couple of bacteria that is low in type 2 diabetes. Could we also match optimal medication based on the microbiome. That's something that is gaining attraction, and especially in the space of cancer treatment, of the new anti-PD-1, for example, these immunotherapies, we seem to be having a responders and non-responders based on the microbiome. But as I alluded to in the beginning of the talk, we also have a big interest in understanding, do the microbiome produce molecules that can contribute directly to insulin sensitivity? And this would be a way to understand the mechanisms, but if we understand the mechanism, perhaps we can start to use these molecules either to add them if they're beneficial or inhibit their production if they're detrimental for insulin sensitivity. So our hypothesis was that type 2 diabetes associated microbiota can contribute to insulin resistance by producing metabolites regulating insulin signaling. So we addressed this in a collaboration with many different researchers, but the clinical side was led by Max Nudorp at the Amsterdam Medical Center, where we had the opportunity to get plasma samples not only from the periphery, but also from the portal vein. And this is a vein going from the intestine to the liver to really study microbially produced metabolites. And the reason why we could get that is that at the AMC in Amsterdam, they're performing, as they do elsewhere, many different bariatric surgery procedures in obese individuals. And Max and his colleagues managed to get us a sample from those, from 10 individuals without type 2 diabetes and 5 with type 2 diabetes. We then performed metabolomics, which basically means that we measure molecules in the plasma samples, and we do that commercially at a company called Metabolon. And since we had relatively few individuals, we couldn't study all kinds of metabolites, but we focused on amino acid-derived metabolites. The reason why we did this is that amino acid-derived metabolites are very important signaling molecules. You may have heard of serotonin, which is a tryptophan metabolite. So we think that let's do something new and look at that set of metabolites. And we identified four different metabolites that were more abundant in subjects with type 2 diabetes compared to subjects without type 2 diabetes. So as a microbiology lab, we were interested in those that had a microbial origin. And how can we filter out those? So we did a similar experiment, but now we used mice that were either germ-free without any bacteria or conventional raised with bacteria. We took portal vein samples and also from the periphery, and unfortunately we couldn't find dopamine sulfate nor N-acetylputrescine in these mouse samples. Glutamate did not really differ between germ-free and conventional raised mice, but imidazopropionate was significantly elevated in conventional raised mice compared to germ-free counterparts, both in the portal vein and in the periphery. So this is, of course, very good news in the sense that it's a microbially produced metabolite, but it also shows that we did not have to go through the ordeal of having portal vein samples. We could have done it in peripheral samples as well. So from where does imidazopropionate arise? So this is like the canonical metabolism of histidine to glutamate. Our cells and the bacterial cells have this pathway, but the bacteria also have another enzyme that can take this intermediate urocanate. And instead of using urocanate hydratase, they have a urocanate reductase, and the urocanate reductase will give rise to imidazopropionate. And what Tanvir Khan did, who's as a researcher in the group, he has developed a bioreactor where we can make a artificial gut that we can colonize with a fecal sample from either a subject with type 2 diabetes or a healthy control. We let it stabilize for a week, and then we put in the substrate histidine, and we measure imidazopropionate for the next 7 hours. And what we found was that subjects without type 2 diabetes, they did not really produce any imidazopropionate in this time frame, whereas those with type 2 diabetes, they produce significant amounts. So I think it's fair to say that the imidazopropionate is a microbially produced metabolite, and that the microbiome in subjects with type 2 diabetes appears to have a high propensity of producing this metabolite. And we have been able to validate that in a large European consortium that we've been a part of called the Metacardis. Marcus Henriksson did a lot of measurements, and we could then see or confirm that in this cohort where we have Danes, French, and Germans, that type 2 diabetes individuals, as well as those with prediabetes, have significantly higher levels of imidazopropionate than healthy controls. We can also then do what we call an odds ratios, basically saying if you are in the fourth quarter, so if we divide all these into 4 groups, and that group with the highest imidazopropionate levels, they have about a 2-fold higher risk of being in the prediabetes group, or even a 3-fold higher risk of being in the type 2 diabetes group. Interestingly, what Antonio and Pierre, who did this analysis, has found was that the histidine intake did not affect the imidazopropionate levels. So if you eat a lot or a little histidine, does not seem to be important. Most likely because the histidine and most of proteins are digested further up in the intestine. So what they instead found was that the microbiota seems to be altering type 2 diabetes or at those with a risk, and then metabolizing histidine differently. So we found that those with a lower gene count, so with fewer microbial organisms, had a higher levels of imidazopropionate. And this was true if we looked at healthy individuals, those with prediabetes, and those with type 2 diabetes. And more interestingly is that bacteria that we see are increased in type 2 diabetes, they were positively associated with imidazopropionate, suggesting that they may be the source of this metabolite. And a bacterium that is negatively associated with imidazopropionate is reduced in type 2 diabetes. So we're quite excited about this, that the altered microorganisms in type 2 diabetes may having an increased propensity of producing imidazopropionate. But what does my imidazopropionate produce insulin resistance? Well, the answer is yes. Antonio has been injecting germ-free mice with imidazopropionate and can show that they do not tolerate and clear glucose as well as those that are controls. Our co-host was a very talented postdoc at the time, and now a professor at Postec in Korea, has been really trying to investigate the molecular mechanisms of imidazopropionate signaling. So this is a very simplified version of insulin signaling. Insulin binds to the insulin receptor that is recruiting insulin receptor substrate 1 and 2 that get tyrosine phosphorylated, leading to AKT activation. But if IRS1 and 2 instead is serum phosphorylated, it will inhibit the tyrosine phosphorylation, and it will also target the IRS1 and 2 for degradation by the proteasome. This will, of course, then inhibit AKT signaling and thus insulin signaling. So what Ara did, she was taking the samples that Antonio had obtained from germ-free mice, either treated with vehicle or imidazopropionate, and she did a lot of immunoblotting. And as expected, if you just have been treated by vehicle, you have the insulin receptor, you have the insulin receptor 1 and insulin receptor 2 in the liver. But mice treated with imidazopropionate, they had normal levels of the insulin receptor, but they had reduced levels of IRS1 and IRS2. So in the germ-free mice, this is very black and white. In conventional mice with bacteria, we see a clear reduction, but it's not as obvious. But we also wanted to see whether imidazopropionate was acting in a cell autonomous way. And we did that by taking hepatocytes, so liver cells from conventional raised mice, and we cultured them in dishes, and we treated them for 20 hours with imidazopropionate or with vehicle. And when Ara was immunoblotting them, we saw a nice reduction in both IRS1 and IRS2. And as you can expect, if you have been treated with imidazopropionate, the cells do not respond to insulin by AKT phosphorylation as they do in the controls. This is a physical or biological physiological readout for insulin resistance in these cells. So to make a very long story short, what Ara did was that she really mapped the signaling from imidazopropionate. And if you're interested, you should read this paper here that she published a few years back, where she finds that imidazopropionate activates a MAP kinase, a protein in the cell. We don't know how it enters the cell, but somehow it seems to do it, where it induces autophosphorylation. It do it mainly on the p38 gamma, but p38 delta seems to have some effect. The p38 gamma then induces phosphorylation of a protein called p62, leading to mTORC activation, S6K1 phosphorylation, that is then responsible for the serine phosphorylation of RS1. So this is all great. So this is mainly done in cells and mice, but do we see any of these correlations in humans? So we were lucky that in the sample set that we got from Max and from Amsterdam, they didn't not only take the polymerase plasma, they also got liver biopsies from the patients. And by performing immunoblotting, Ara could see that the signaling pathways that were induced by imidazopropionate, such as P62 and S6K1 phosphorylation, we found were more induced in subjects with type 2 diabetes than in normal controls. This was also associated with at least a partial reduction in IRS2. So we think we have here a molecule that is microbially produced and may contribute to insulin resistance in humans. So we think this is also exciting from a different perspective, and this is a future aim of our research here. Can we find means to change the microbiota or even inhibit urocanate reductase to reduce the levels of imidazopropionate and thus improve metabolism? So with that, I would just like to thank you for listening and also thank the people in the group. It's a lot of people being involved in this work, but particularly I would like to highlight Göran Bergström and his team, Valentina and Tony, who performed the analysis of the microbiomes, Antonio and Ara, together with Max and his team and the Metacardis team, that been really responsible for developing the imidazopropionate study. And this is how we look like. This is from a recent photo when we were hiking out in the forest behind the university here. And with that, I will be happy to take your questions. So thank you very much.
Great presentation. Thank you so much, Fredrik. Really, really interesting. And I can see that right now there are already several questions popping up. So I will start with some of them, with them actually. And before starting, please, if you have questions, keep writing them. So I will start. The first question that we have is, has the oral microbiome an impact on the gut microbiome?
Oh, this is a great question. Been a lot of debate around it, and we see and others see signals of oral microbes in several metabolic diseases. The first one was cirrhosis, which is a severe liver disease, and we think that there may be mechanisms that enrich these, and we don't know if it's direct or that several of the patients may be on proton pump inhibitors. So that lowers the pH and allows more of the oral microbes to come into the gut. We think that there may be additional mechanisms that may actually be even contributing to that disease. So for example, we see a group of bacteria called Prevotella that seem to be changing from the gut associated ones to the oral ones in certain diseases. So that's one of the notions. The other is that there's another bacteria called Fusobacteria nucleatum, and that's also oral derived, and seems to be associated with colon cancer. So yes, there seems to be a link between the oral and gut microbiome.
quite interesting. And I guess, I don't know if it was expected or if that would have been what everyone was actually hoping or looking for. Another of the questions that popped up is, would an inhibitor to imidazole propionate synthesis help reduce type 2 diabetes, or is it less of a causal relationship and more of an indicator of type 2 diabetes risk? They refer like, okay, this has been mentioned in the Cell 2018 paper, but I don't know if you would like to elaborate on this.
Yeah, so I think it's a once again a very good question. So I don't think that blocking imidazole propionate will treat all diabetes in the world, but there may be, and as you saw, that there was also an overlap between those with and without type 2 diabetes and levels of imidazole propionate. Some had very high levels, some did not. So I think what we will come to is that type 2 diabetes, as you know, has several different etiologies, and maybe some of them is a subgroup where you have high imidazole propionate. So targeting those, so I would say maybe as a more like a compound diagnostics, that you need to measure imidazole propionate, and then you find those individuals. What we have to see is whether you can prevent the disease or if you can treat the disease. Maybe you have to identify them before they have a disease. But that's a long ways to go, but it's something that I think is very interesting, and I think we have to move in that direction also from a scientific point of view to really establish causality in humans.
Absolutely. Another question is, there is no correlation with the levels of histidine in the diet?
No, we can't find any. So we think that that's, you know, a lot of the food supplements for athletes contain a lot of histidine, and I don't think those will lead necessarily to high imidazole propionate, because most of that will be taken up in the small intestine. So we will not see it. So it's really the histidine in the colon, and some of it may be microbially dietary derived, but we can't pick up that signal. We think a lot of it may come also from like cells on the epithelial linings that then get sloughed off and then can be a protein substrate.
Then there are several, of course. It's great to see this. You focused in on an effect of on insulin resistance. Any thoughts on the effect of insulin secretion by microbial metabolites?
Yeah, we in general, when we have done our experiments, we don't see so much on insulin secretion. That said, there's a lot of interest in how microbial metabolites, including short-chain fatty acids and bile acids, can activate TGR5, which is a GPCR, and lead to GLP-1 release, which in that turn will lead to insulin release, or insulin secretion. But in our hands, we see the main effects on insulin resistance.
Okay. And do you know if there are any differences in energy intake or expenditure in response to IMP treatment in vivo?
We have to investigate that formally, but we don't see major effects on overall energy expenditure. It's something we would like to do in more detail.
I see. And then, yeah, as I said, like, I think there are people who has been really interested, and they have so many questions on. There's another one here that says, how can these micro products pass from the gut to the tissues that you mentioned? Would it be expected, I don't know, by the lymphatics or bloodstream?
So once again, it's a very good question. There's not a single question for them, no single answer for them. I think that for like endotoxin lipopolysaccharide, which has been sort of a very hot topic, in my view, that will be transported together with lipids and then being transported in the form of chylomicrons, most likely. Other metabolites like bile acids will be having an active transport from ASBT in the small in the lower small intestine, and then a passive transport most likely in the colon, and being then transported through the portal vein into the liver, in a enterohepatic circulation where some spillover will reach the periphery. Imidazopropionate, we think it's a small molecule. It's it's amino acid derived, so it may or may not require a specific transporter. My bet would be that it's a histone transporter, amino acid transporter. We haven't tested that yet, and then that would be transported also in the portal vein. Other lipid-mediated molecules may be then in form of chyme and go through the lymphatics. So I think that's several different possibilities. So you have to look at specific metabolites in order to give that response.
Then, what are the microbiome strategies you want to take to decrease the IMP? I don't know if that would be too specific.
So we have several that we are talking about, but that's everything from seeing if we can increase the microbial diversity and changing the environment in the gut by that way, and that would then supposedly reduce the number of IMP producing bacteria. But we also would like to see if we could block urocanate reductase directly by finding inhibitors.
There comes, of course, more other questions more related to type 2 diabetes. When you study type 2 diabetes associated microbiota, is it necessary to exclude pre-diabetes cases, or what would be your perspective on this?
I think that for me, I'm not a clinician, but what is puzzling or fascinating to me is that clinicians really like to have a cutoff. And no matter how much or how little you're above that cutoff, you are diseased. And it doesn't really matter how far below you are, you're healthy. And a lot of these disease biomarkers, to me, are continuous. So I think we need to be better at really defining the patient populations and the groups we're looking at, both from potentially knowing more about the etiology, but also then to see whether there's overlaps, and maybe some are just further down in the disease progression and others are going in that direction, and that's what we may be seeing. So I think the interesting part with the pre-diabetes patients or individuals, it may say that there's actually a chance that they contribute to disease and not just reflecting the disease. And maybe we can also then find windows where that improve possibility to prevent the disease from really becoming full-blown overt diabetes.
How malleable is the microbiome colonization after early life gut microbiome seeding?
Oh, these are really good questions. There's, it's a big debate in the field. At least what I think is it's a little bit like the glass half full or half empty, and I think we don't know sufficiently much about the microbiome, to be honest. So what we are trying to do now is just to get estimates for how variable is the microbiome in an individual over a year. So what can we expect beyond and above that level? And what we find preliminary is that it's individual specific. Some individuals have a very stable microbiome, others have a very easy to change microbiome, and within this, there's certain bacteria that seems to be more stable than others. So it's a complex question. What we do see is that it seems to be hard to find consistent changes over individuals. Many of the responses probably would be individual if they're in sort of the normal range when it comes to diet, except if you remove fibers completely, then you have a completely altered microbiome relatively rapidly. When it comes to medications, metformin, for example, has a huge effect on the microbiome. That's easy to pick up. You restructure the microbiome, and it seems that restructuring, at least to some extent, seems to be contributing to the clinical effect of metformin.
Following up with this question, do you know if there would be any sort of relationship between how stable the microbiome would be, for example, depending on gender or depending on the age of the patients?
The gender, we don't really know if we don't see a strong difference between genders when it comes to stability, but age, yes, it seems like that we have we have a very changing microbiome early in life and also in late in life, that you sort of, the stability falls apart in general. So it's sort of like you go from chaos, you get stable, and then you could become more chaotic again, to simplify it.
Yeah. There's another question here that says, Faecalibacterium prosnitzi is inversely correlated with imidazole propionate. Since this one is one of the main butyrate-producing strains, could be attributed to higher butyrate levels.
Yeah, it could be. I think one of our sort of key hypotheses right now is that Faecalibacterium prosnitzi is a marker for a complex microbiome, a healthy microbiome, and also happens to be a butyrate producers. I don't know if I would dare to say that butyrate and imidazole propionate is directly connected, but maybe if you have a more stable and well-functioning microbiome, you have less room for the imidazole propionate producing bacteria. So potential by increasing the net levels of Faecalibacterium prosnitzi, you could change the microbiome and then affect the imidazole propionate production.
And we have another, I think that people overall has been pleased, extremely happy with your talking. They keep asking more stuff, like is it known that it is known that metabolic syndrome is associated with inflammation. Have you checked if imidazole propionate can reduce this metaflammation?
So imidazole propionate seems to be actually contributing to inflammation. So that's how it was identified in the 1970s, that it was associated with inflammatory bowel disease. So I think, and p-perry gamma is a MAP kinase that is well known to be involved in inflammation. So our idea is that the imidazole propionate will then increase the inflammation.
Then what happens with the microbiome and imidazole propionate when you reduce weight after bariatric surgery?
So there's different kinds of bariatric surgery, but Roux-en-Y gastric bypass is probably the most well studied so far. And what we tend to see there is that of course, it's also, bariatric surgery is one thing, but you also change your lifestyle after bariatric surgery. So we see an reduced capacity to digest carbohydrates, we think, or fiber. So we think one of the things that's happening is that you don't accumulate as much energy. That has to be substantiated in larger studies. But we also see an expansion of E. coli and other proteobacteria at the level of oxygen tolerant. We don't see much regulation of IMP after bariatric surgery, to be honest, the studies we have performed, neither in sleeve nor in gastric bypass.
Here's another one that actually they are asking, like, have you investigated about the crosstalk between the gut microbiota and central nervous system regarding dietary regulation?
We haven't. There's a lot of interest in the field, and especially signaling through the vagal nerve. We have been contributing to some studies where we have seen that there seems to be some crosstalk between what's called intestinal gluconeogenesis. So a lot of the short chain fatty acids can either regulate or become a substrate for glucose production in the intestine, and that newly synthesized glucose can then signal to the host from the portal arch through the vagus. There's also some, since the microbiome seems to be regulating GLP-1, which also is a neurotransmitter, we think that there definitely are links. We don't understand them. We do see effects on appetite, and we have seen that the microbiome can affect then hypothalamic inflammation that could be potentially being associated with leptin resistance. So there, and many other people are much more into this than we are. So I think that we will learn a lot. I think one of the issues here is how to translate what you find in animal studies into humans, but that's not unique for that branch of the research. It's for many.
I see.
Especially when it comes to behavior.
So I beg your pardon?
Especially when it comes to behavior, that mice may behave differently from humans.
Yeah. And then here we have another question that is, you mentioned that histidine does not affect IMP. You mentioned that histidine does not affect IMP levels, but may it be other precursors from diet?
No, so maybe I've been unclear there. So I think dietary histidine does not seem to be correlated with nosopropionate. Histidine is the precursor for IMP. It's just that what we eat in terms of histidine, we don't see a strong correlation or any correlation with IMP production.
Okay. This is a fascinating talk. I mean, you're being congratulated for your talk by everyone. And it says, I was interested in knowing the following. Did you find any correlation of butyrate production with the levels of amino acid derived metabolites that you studied? 1. And 2, also, did you find interaction levels of other amino acid derivatives apart from IMP in human studies?
Yeah, so the ones that we have mainly been seeing, and if I ask the second question first, or answer the second question first, was this what I showed, that glutamate in type 2 diabetes. We also saw this dopamine sulfate and N-acetylpyridine in our study. We have seen also in some other studies, like branched chain amino acids, for example, which Chris Newgard showed probably 10, 15 years ago. So there seems to be a slightly altered microbial metabolism on amino acids. And the butyrate, I think, is harder to respond to because we have problems measuring butyrate in humans. From that what we would measure in the feces or the serum, we don't know if it's a true production or if it's differences in absorption by the host, or then used as a subject in the colonocyte. So if you measure something in the feces, is that reduced utilization or uptake of the butyrate, or is it increased production? The same thing goes for plasma, where we have problems of measuring butyrate. Some people have been successful in measuring butyrate in the circulation. We have not, when we do all the controls, that we have a lot of background and have a hard time to get a signal because the levels are low. What we do know is that healthy microbiome prefer digesting and metabolizing fibers, which would generate more butyrate. And if you don't have sufficient amount of fiber, you go to putrefaction or fermentation of proteins. So I think there is an inverse correlation, but we have a hard time to prove it in humans.
See. And then it says, despite butyrate producers, did you also find correlation between type 2 diabetes and the levels of butyrate in stool or blood?
No, we haven't measured butyrate of that given reason.
Then they said, like, since you observe correlations between F-prosnitzi and imidazole propionate, have you evaluated if addition of F-pros could influence the levels of imidazole propionate?
That's once again a very good question. We cannot do that in mice. We have problems of getting F-pro to colonize mice, especially the strains we have isolated. So we haven't really been investigating that. As I alluded to, we started to do interventions with Faecalibacterium prosnitzi in humans. That's one of the things we would like to measure, of course, but we don't have the results yet.
We have here, I think, a couple more questions. There's one that they're asking, like, I have read that children have similar microbiomes as their mothers. Is that true?
So what we tend to find is that after a vaginal birth, when the child is born with his or her nose and mouth towards the mother's anus, they have the initial seeding from the mother. We can also identify bacterial strains from the mother later in life. But of course, when you start to eat a lot of breast milk, the microbiome composition is very different from a human, from the mother who eats solid foods. But the absolutely first sample, yes, we can see it's similar to the mother.
Well, I guess, I mean, there are still quite a lot of questions, but think about the time that you have already spent with us, and we're very thankful for this amazing presentation. And again, we will send you all the questions that the people has sent, all these attendees, a lot of attendees. We are very pleased to have you again. Thank you very much, Professor Bäckhed. to our attendees, please, you can contact us and we'll try to find the answer to your questions. And again, thank you for joining us today. Thank you for the presentation, Professor Bäckhed. And we will be happy to have you again here. Who knows, maybe we'll have you for another presentation or a discussion panel. And to all our attendees, again, thank you so much. Please do not hesitate in contacting us if you have any questions or if you want more information. And we will end up for today with this webinar. Thank you so much to everyone.
Thank you.
And have a nice day.